It is our pleasure to present Nicolas Langlade, Dr. Nicolas Langlade from the National Institute of Agronomical Research in France. Thank you for accepting the invitation. And I will give a short presentation before your talk. Nicolas es investigador del INRA. Él hace más de 10 años que está trabajando en girasol, en el área de genética, genómica y fenotipado. Es líder y director de investigaciones del grupo de genética y genómica de girasol, en una división que es de biología vegetal y mejoramiento. Ha tenido una participación esencial en lo que es la generación de una referencia genómica de girasol, que es la que muchos de nosotros usamos y que ha potenciado mucho la investigación de girasol desde que salió en 2017 hasta ahora. Nicolás además ha participado activamente en la generación de herramientas para que todos los investigadores y la comunidad, tanto académica como productiva, acceda a esa información y se pueda aplicar en el mejoramiento del cultivo. Él desde 2012 también lidera una iniciativa público-privada de investigación para lograr mejorar y estabilizar el rendimiento de girasol en situaciones de estrés hídrico, y hoy nos va a dar dos charlas. La primera tiene que ver con ese proyecto que es de participación público-privada. Va a dar un overview de cómo es el manejo y la interacción, y en una segunda parte de esta charla va a dar resultados, va a hablar de resultados alcanzados en el área de estrés hídrico con una aproximación de predicción genómica, que es una de las cosas que nos interesa mucho para el mejoramiento de girasol en Argentina, y en lo cual todavía estamos en muchos casos, tanto en la parte pública como privada, arrancando. Bueno, entonces, welcome.
Thank you, Norma, for this introduction. As Norma presented, I'm very much involved into public-private partnerships in France. It's a long time effort for the public academic community on sunflower in France to collaborate very tightly with private companies, so breeders, etc. And when I was recruited at INRA 10 years ago, I took in charge this animation at different levels. And here, at the beginning of my talk, I will present you what we do in a large genomic project named Sunrise. And then after, as Norma said, I will focus on more scientific point that illustrates a little bit what we can do with this kind of public-private projects. So, first thing, I'm going very quickly on sunflower. Of course, you all know the crop. It's a very important European crop. On a continental point of view, Europe is producing almost 80% of the sunflower production. It's also important in EU, and so mainly with France, Romania, Spain, Bulgaria, Hungary, etc. And so we are very interested in maintaining this crop that has a very high ecological friendly aspect. So in the context of climate change, of course, there has been several studies already published showing that sunflower might be very much impacted, because basically it's oilseed crop for 1,000 countries around Mediterranean Sea, and this is where in Europe climate change is going to be the most dramatic, and France will be really impacted as well. And so in this study in 2010, Moriondo actually calculated that the impact could be up to minus 20% if nothing is done in matter of varieties, new varieties, and new cropping management. And in Southern Europe, like in Greece, it's part of Europe the most impacted. It will be much more important, and actually maybe sunflower wouldn't be worth growing over there if nothing is done. What we've done in the Sunrise project, and I will explain this later in the second part of my talk, we actually showed that 1 day of stress, of drought stress in France on our multi-environmental trial, so meaning the stress there are less than 60% of available water to the crop, actually impacts the yield at 0.4 quintal per hectare per day of stress. And in our trials, we tend to have 30, 40, 50 days of stress. So when you actually add up this to the actual cropping area in France, the impact of 1 day of stress is a matter of millions of euros that are not in the farmer's income. It's not for breeders or so on. It's really the impact on the farmer's income. And so this is a very motivating point also to find varieties that are more adapted to drought stress. And when we added up at the whole year level with all the days of stress, it's much more. And if we think that there is about 25 million hectares of sunflower, and here we're only talking about very small producing country, France, the impact of drought stress for sunflower is major. We are not going to remove drought stress, obviously, with climate change, but what we can do is actually produce knowledge, produce tools, ways to make this impact less important economically and also on the production, because we need to maintain or improve the global production of food in general. So this is all this motivation that actually led the French community to gather in basically 2010, 2011 and start a major project. It was a very unique way of organizing this. There was a huge carrot, so everybody gathered. A lot of political pressure also for the breeding companies to join. This is an 8-year project from 2012, and it's going to finish next year. The total budget of the project is 21 million euros. But it's 1/3 is funded directly by the companies, and they have tax credit, etc. 1/3 is funded by infrastructures and human resources of the public partners, and 1/3 is actually direct funding to mostly academic partners, but also direct funding to the companies. And it gathers 16 partners, public and private. So, one thing, here's a map a bit of where are the partners. There are different categories of partners, but one thing I can stress already is that actually most of the partners, nearly all the partners, they are based in Toulouse. And Toulouse is the most important sunflower production area in France. So basically, when we go to work, when we do everything in the day life, we see the sunflower seed fields around us. And this is actually very motivating. And also, we know many things on normal life of sunflower farmers. So this is, I think, a very positive aspect that all this community is gathered, and so we can also meet informally very often. So seed industry partners, basically you have, well, it was Biogema, and now it's called Inolea. So it's a biotech company, and 5 sunflower breeders or a joint venture for Limagrain and Euralis, which is named Soltis. It's for sunflower research. And then COSAD, who I don't think is present in Argentina, Mass Seeds, I'm not sure either, but RGT, Limagrain, Euralis, and Syngenta are, of course, major breeders here. And the Technical Institute, Terinovia, so it's to give advice to farmers, to evaluate also some new varieties, new cropping system, etc. So it's really applied and in direct contact with the farmers' development of research findings. And then at the academic level, there is a multidisciplinary public research center in Toulouse, joining agronomists, geneticists, bioinformaticians, mathematicians, and also platforms, technical platforms for genomics, so sequencing, genotyping, etc., and phenotyping platforms. And I will I will show a little bit of example in this talk, and I have a second talk later this afternoon, and I will go a bit more in depth into the phenotyping platforms. And they are based in Toulouse, in Bordeaux, and in Paris. Okay, so what I wanted to do was kind of highlight also how the project is organized a little bit, some key results, and also a point of view on things that were brought to breeders, since the major private partners here are breeders. So basically, the project has a board, like with one representative per partner, so 16, and it meets at least twice a year. There's a scientific committee with work package leaders that are both public and private. So, and this is very important to have public, private involvement very strongly at the scientific level, because private scientists have expertise that are not present necessarily in academy, and so this is really helpful to have them. This is a meeting very often, like 4 times a year. And then we have 10 work packages, so management, genetic resources development, genomic resources development, phenotyping, crop modeling, genetic discovery and validation, bioinformatics, socioeconomics, I will come this later, former project validation, so for we add enabling technologies from other projects, and so it's a separated intellectual property, so it's organized this way. And then dissemination, communication, training for students, etc. is another work package. There is an external international scientific advisory board from national and international experts, and including Ruth Hines here, and from INTA, and people from Canada, Italy, USA, and this meets every 2 years. We didn't use it much during our project, but I think this was maybe a mistake. I know other projects of this kind, because there are this kind of project for the main French crops like wheat, maize, rapeseed, sugar beet. And I know in the other project they used it much more, the scientific advisory board, and I think it was a good idea. And there is an IP committee, because all this all the knowledge, the tools, etc. produced are under IP very strict regulation, and so there are lawyers from the private, from public-private company, and from the scientific board, and all the IP is basically based on what each partner actually justified has done in the project. So so this also brings them to get involved and keep their engagement until the end. And okay, that's all I want to say about this kind of organization. On the management, we have a permanent project manager that is helping me running the project, because with 16 partners there is a lot in fact happening, and the project manager is also in charge of some dissemination, doing the movies, newsletter, etc., organizing annual meetings. Every year we organize annual meetings with about 70% from public and private. We have half-year working meetings, newsletter, internal and external, a Twitter account, a website, and everything is actually managed by this project manager. And so now that you have an overview of this project, how it's organized, I can explain you a little bit what it's mainly focused on. So the two major focus of the project are developing hybrids that are tolerant to drought. And in fact, to do this, we need to understand, on one hand, drought tolerance at the genetic and the molecular level in both controlled and field conditions, and a drought as a part of a complex of different abiotic stresses. Drought is never happening itself on its own. It's always linked to nutrient deficiency. It's in the environment. It's correlated to cold condition. When you go north, you have colder condition in the early, but it's wetter, so in fact it's not really independent in the multi-environmental trial. So this is something that we need to take into account. And on the other hand, this needs to be applied in hybrids. And in fact, in the hybrids, to predict the behavior of a hybrid is actually very complicated. It's not just the sum or the mean of the two parents. And so that's why we had a second goal also, to understand heterosis better and be able to predict these traits in the context of heterosis, but based on information also on the lines, on the parental lines. So this is why we wanted also to address as much as we could the heterosis at the genetic and molecular level in both conditions. And for this, we have basically three scientific axes. So let's say strategies that are complementary. The genomics and systems biology approach, so it's basically developing knowledge at the genomic level, sequencing the genome, describing how genes function, interact together, etc. Combined genetics and crop modeling as approach, so it's really being able to predict complex traits such as tolerance, yield, based on genetic information, using quantitative genetics, but also predicted in environments that were not experimented. And so it's the case with climate change. Future environments have a very low frequency nowadays, so we are unlikely to have experienced them. And so, so we need to have models that actually make simulation in new environments and also with new management practices. So this is this combination. It's one scientific strategy. And another scientific axis that is much more reduced in the project, and I'm not going to discuss this much, I'm not a specialist either. It's the socioeconomics of sunflower production in general, and registration of the of the varieties. Okay, so now just want to highlight some key results for this first scientific axis, the genomics and systems biology. So sometimes in 2014 we developed genotyping chips based on genome resequencing. We didn't have a proper genome reference, but we were still able to develop these genotyping chips, and they were really helpful throughout the project, but also as soon as they were produced, they were incorporated by the sunflower breeders and for their breeding programs, and this was a very applied and key tool that was developed within the project. It helped developing genetic resources. I will come back later. And also, it was helpful to build new projects that were both academic and public and private projects on orobanki and genetic resources in sunflower. In 2016, we produced the first sunflower genome. This was really thanks to this large project. We were able to actually assure the funding and the use of this of a new generation of sequencer, the PacBio sequencers, and because there was this size, this organization, etc. When we all together, the public and the private, we tell the institutions, the state, or the region, that it is a key infrastructure, a key equipment for the community, the sunflower community. It has a very much weight, and actually it's helping politically to be organized like that for long-term and large projects. And this is what happened with the sunflower genome. Thanks to this organization, we're able to actually get new money from the region, buy a sequencer, and actually unlock a major result to produce the sunflower genome. And this sunflower genome, of course, we were able to describe on a genomic point of view the breeding history of sunflower. Thanks to it, it was already as soon as it was produced, incorporated into by the breeders to accelerate their breeding process, position the markers on the genome, etc., with much higher precision. And it helped also developing new research projects. And then it also, because there was a breakthrough in technology at different levels, we did a patent application for sequencing this. It helped other major projects on maize, especially for genomic sequences, and also on the parasitic plant Orobanchi. So using the same technology, the same approach, we're able to sequence the Orobanchi genome. And this was published in 2017 and released to the general community. And then where in this paper we described all the oil pathways for sunflowers. So basically all candidate genes that control oleic, linoleic, composition, tocopherol, etc. Were described in this paper. We also identify in the paleogenomic level, so this is the history of the evolution of the genome, that in the past, 40 million years ago, the genome duplicated, so every gene was present in 2 copies. And since then, the genome is losing some copies and returning to a normal state where there is 1 gene. And this was, and so we were able to describe this, and this is actually very helpful for breeding, because if you have 2 genes that are very similar and have the same function, you need to actually have markers specific to the 2 genes and breed using this information. And this is very different from having only 1 gene controlling 1 trait. And we also describe the gene networks controlling flowering time in sunflower, and obviously flowering time is a key trait for breeding to control the cycle length, etc. And to adapt sunflower to new cropping system, etc. It is very important to know this gene network. And of course, since then, the genomics continue on sunflower, and now we collaborated with Vancouver in Canada, a team led by Lauren Wisberg, on the description of the sunflower pangenome. So the pangenome is actually a description of all the genes that are present in a species. And so this helped this description. On the socioeconomics point of view, I'm going to be quite fast on this one. Our work was basically focused on the impact of the policies, so on the choice of farmers to go for sunflower or other crops, generally to choose for the cropping system. To describe the industrial chain and the valorization of oil cake in the context of sustainable development, and also define the concept of an eco-innovation. So basically it's a technical innovation that is helping on an ecological point of view. So it could be a genetic marker that is actually making the crop produce with the same with less water, for instance. But it could be also a new way to speed up breeding and save time and efforts, and then have an ecological positive impact. So this concept was actually developed with the example of the sunflower breeding in the context of sunrise. And we also, with the policies, this was done in France, in Hungary, Bulgaria, and Romania, so at a continental level. And we also studied the pollination of sunflower and how sunflower can help this ecosystemic service. And also the impact of the policies, basically, and the choice of farmers for their incomes. Now, the third point, and the one I'm going to talk also later on, is the integration of genetics and crop modeling. So, on one hand, we developed phenotyping tool to help the description of the crop and how plants develop and respond to stress. And this was done by the development of the Eliafen platform, development of 3D models of new projects. We developed also a UAV, so drones, in collaboration with another large project named Phenome, and the Phenomobile. And I will describe this quite a lot this afternoon. And one very important result was actually a new method to model the stress and how they interact. And this stress modeling is actually helping with the breeding in the context of genotype by environment interaction. And thanks to this, we were able to identify molecular markers, SNPs, that were correlated to drought tolerance, to flowering time, to oil content, and the response of these traits to drought stress. And this is now we are in the frame of the collaboration at the international level with teams in Argentina and in America as well, and develop new genetic material based on these results. This was published, and one very important result for coming from the Sunrise project is actually the first genomic prediction models for sunflower. So based on the genomic sequence, we're able to train models and actually predict the phenotype of new genotypes that are not described yet. And so this can be this can help the selection of interesting genotypes, and then we can go on the phenotyping or the breeding. And this was published in Frontiers in Plant Science. Okay, so now I wanted to maybe focus on some tools that were maybe of interest to breeders, and one of it is actually the production of genetic resources. And this is very important to introduce novelty, genetic novelty in the breeding programs. So this is really at the level of pre-breeding, and this is where the public-private collaboration is very useful, because this is pre-competitive. We can share resources, etc. And then when things are becoming very interesting to breeders, then they internalize the breeding process. But beforehand, there was a common effort to create some genetic diversity that is really essential to adapt sunflower in the future to the different challenges. And so one strategy was, so here are two different populations that we used to discover how genomic regions control drought tolerance. It's an incomplete factorial hybrid panel and a NAM population. So an incomplete factorial panel is actually a cross between, you have 36 males, 36 female lines, and you don't cross all of them, because it would be, in this case, 1,296 hybrids, and it's a lot and quite expensive to do. But we crossed 1/3, basically. So this was 1/3 of the hybrids were produced. The original parents, it's a mix of private lanes and public lanes. So it's 10 female and 26 from the private, and 26 females from the public lines, 5 restorer lines from the private, and 31 from the public. And so everything is crossed. 1/3 of the crosses are performed, and we are able then to phenotype this panel, and thanks to genotyping by resequencing. So we resequenced all the 72 lines 10 times, which means that we are able to identify millions of SNPs, and after some filters, we actually work with SNPs that have a unique genotype, so a unique pattern, if you want, among the 72 lines. And this means for 400,000 SNPs that are used then for genomic prediction, genome-wide association mapping, etc., to discover how this genomic region controls the trait of interest. So this was one genetic resource that was produced. It's an original design, and then I'm glad to say that afterwards it was adopted by maize and maybe improved a little bit by the maize community in France. But it turns out to be very effective, because you have in one generation you already have your diversity panel, and you're in hybrids. And this is very efficient also on the cost of resequencing, because you don't resequence 400 lines, you resequence only the 72 parents. The second genetic design that we used is a NAM population. So a NAM population, it's a nested associated mapping population. It's basically taking 9 parents here with these orange circles, and each parent is crossed 4 times. And this is in double round robin, so you cross it with the neighbors in this circle diagram. And then from each cross, you generate a recombinant inbred line population, but of a limited size. So here we targeted 32 lines. In fact, we produced between 100 and 200, but then we only genotype and phenotype 32. And then they become 18 real populations. So it's about 500, so this is it. We had 9 R and B public lines, so only public material. 5 they were served until the F5 or F6, and genotyped using one of these genotyping tools I mentioned earlier. And actually at the end we work with 100,000 SNPs that really saturate all the position in the genome, and so we can identify where the crossing over happened. And this 572 recombinant lines were evaluated for phenotyping on 3 testers, so we could actually also study some heterosis effect using this genetic design. And at the end we have 1,500 hybrids that were characterized in a multi-environmental network. So it's a very large population. It's the largest effort we've done in the project. And we are currently in the middle of the genetic analysis. So I can't tell much about results on this. Okay, so all this was phenotyped for yield, oil content, also in some environments for architecture, so it's leaf number, height, position of the largest leaf, diameter of the stem, in some environments for the harvest index also, the leaf senescence, and there were molecular biology sampling, so we can also study the gene expression or the metabolite levels in the leaves. So this is a very important efforts, and now we have this facility. Everything is stored. So we had a multi-environmental network of 13 environments. So between 2013 and 15 for this population, and 2015 and 17 for the NAM population. Each time it was mainly done by the private companies. One know-how of breeders and private companies is definitely to produce genetic material and to characterize the genetic material. They do it much better than the public partners, and so really this is the main contribution to the project. And because you see these are very important large population. We wanted on several years. We have to produce the seeds, etc. It's very heavy, and so the private companies were able to do it in France. Some of them in Romania, like ASAGIR and COSAD, and some of them in Chile, in like Syngenta. And the other network is mainly in this part of France, mainly around Toulouse, in the west and in the north of the sunflower growing area. And each time still INRA did a trial using phenotyping platforms. Okay, here I described a little bit the efforts, what was doing each partner for these 2 panels that aimed at the discovery to discover the impact of the genomic region. One other genetic resource that we developed, and I thought it was interesting to describe it, is actually an allelic diversity platform. As I mentioned in the previous panels, we used inbred lines that were already present in the genetic resources. So basically, we didn't introduce much new alleles, new genetic diversity. We recombined some, and this is already producing novelty, but there is no new allele per se. So we wanted to have an effort also to introduce alleles from the wild Elianthus anus into elite material. And this is this task on the allelic diversity platform. We generated a population based on Elianthus anus wild ecotypes. So here is the map of 40 wild Elianthus anus ecotypes that we have in the INRA genetic resources. At INRA we have about 500 wild ecotypes, including anus and other species. And so we selected based on the geographical position of this, origin of these accessions. We selected 40. All of them were resequenced, and the genetic polymorphism were positioned on the genome. And from this, we selected 5 that were at different, different position, geographical position, but also that are part of different phylogenetic groups of Elianthus anus. And from these 5 ecotypes, we crossed them we crossed them to an elite line, the XRQ line that was used for the genome sequencing. We produced a population in greenhouses. So each partner, there are 5 populations because we had 5 breeding companies. Each company developed its own population from one donor. And then here you have the one line is is actually one line also in of the population. And here you have the different markers on the columns, and in green you have if it's a wild allele, and in pink you have it if it's the elite allele. And so each generation the partners genotyped this material and selected the lines that were to have an overlap and to make sure that all the genome from the wild donor is present and in small pieces in the different introgression lines. And after a few generations, this gives this kind of a figure. So if this is for one population, you have the different lines on the horizontal, and on the vertical, the different genetic markers, so position in the genome. And you see that from the about 100 lines, you cover the whole genome, which means that all the genome from the elite is now splitted into 100 inbred lines that we call introgression lines, that are, at this stage, there are back crosses 2 and self 3 times. And this was done for the 5 population, and we have about 100 to up to 150 lines for each population. So this genetic material is very important because it's really introducing these wild alleles into elite material, and then we can find, of course, genes that are responsible for the drought tolerance, but also for disease resistance, etc. Many different traits are possibly present in these donors. And we genotype using this microarray chip all this population, and so we know precisely where the wild genomic pieces are inserted. So this is a resource. So we know that we have these lines, but now really, how do you choose the one that is interesting? So basically, we have the QTR. So we know some genomic regions are important for drought tolerance, let's say. But we also know the sequence of the wild genotypes because we resequence them. So we have SNPs and indels, which are small as gaps in the genomes or insertions or deletions in the genome. But sometimes the genomic regions are completely inverted in large scale, so several kbs or several megabases of genome are missing or inverted. And to characterize this, we also developed optical maps for the parents. So we know that actually this region is very similar to the elite, so it would be easy to integrate into the breeding program. Whereas if there is a major inversion, it's going to be very tricky. Can be interesting on a phenotypic point of view, but for the integration, recombination and overcross will not happen normally, and this is going to be technically difficult. So this is also a descriptive information to use this allelic diversity platform. And we can use it for phenotyping and validating that the wild allele have some effects. And this is what we've done on the sunflower Eliaphane platform. So here you see the plants are growing through time in control or stress conditions for one wild allele, one wild accession and the other wild accession. And you can see that actually this green one, it is going to grow and stop the growth in the stress condition, whereas this blue one, it doesn't really stop the growth in stress condition. So now we also have phenotypic description of the wild donor. And so we can have a hypothesis, and if one region would be more interesting for this donor or the other one, according to what we are looking for. You see here the response for in stress condition here was dramatic. There is about 60 to 70 centimeters difference in height after about 3 weeks of stress. So it's very dramatic. And here there is almost no difference in height between the two conditions. So the two have different ways to respond to drought that could be used now according to the strategy for the breeding. And also on an academic point of view, it's interesting to understand why these two wild ecotypes have different responses. Okay, so these were the three major genetic outputs of the project. One thing that I wanted to stress before finishing on this general view of the sunflower project was the data management. Over 8 years, there were, as you understood, there were a lot of data produced, thousands of trials, etc. We counted that now not everything is in the database, but we already have over 1 million of data points from field phenotypic information. So everything is really necessitates to be really well organized and explained and also transmitted to newcomers, because in fact in 2012 and in now 2/3 of the work package leaders have changed. A lot of people have retired, changed functions in the companies, etc. And so you really need to have this informatic system to just make sure that the data is still available. It's also very helpful to keep the just reports, meeting reports, scientific reports, etc. All the presentation, the working documents that are produced that are referenced for in the future. We need also informatic storage and organization for the sequence, but genomic information is very heavy. It cannot be explored by the normal tools, so this is very important. We developed a phenotyping archive and database dedicated to sunflower that is storing the data points, but also manages intellectual property. If one data is actually produced by some partners in some context, not everybody has access to it. And this needs to be implemented into the information system. That archive generates a digital object identifier, so it's unique object. So then you can cite in the papers and anything, you can cite this document because it already automatically generates it if you want. And then it's the first step towards big data and automatic data processing in the future. That is going to be a major challenge, I think, for breeding, is to, with all the phenotypic data that is coming every year, resequencing, etc., we always want to reprocess data, and then we need to automatize it, and this is this first step, having this organized towards this, so it's a major challenge. And then there is an intellectual property portal that was developed by this joint venture of INRA and private companies, that also help with all the divulgation of the results. And then if you want to use some results in other projects that do not involve the other partners on a private point of view, then everything is sorted, you can apply, and then it's followed up, and so the intellectual property is safe. So basically, yeah, on this presentation of the Sunrise project, and I hope that you're convinced that it's very important to conduct a public-private project, and that is helping on both sides. We have this organization. We develop genomic tools, genetic material, data management systems, etc. And there is a key advantage for both as when they are pre-competitive projects. It doesn't make sense to have a large consortium like that for competitive projects, because all the main sunflower breeders, most of them, they're part of it. But as a pre-competitive investment, you can share the phenotyping networks, the development of genomic tools, etc., that are very expensive. And then after everyone internalize according to its means and objectives the results. And this is, for sure, it's a long-term investment. And so this is why also this long-term project of 8 years is really meaningful in our field. Which is fairly slow, with 1 or 2 experiments per year. Okay, so now let's move on to the second part of the talk. So here I'm going to be to really focus on. 45 minutes? I thought I was. Yeah, okay. So I will go quickly on this one. So we studied this population. So it's basically a core collection with the public and private lines, genetic material, both male and females. And this was done some years ago in the French environmental network over 4 years. And so what we want here is to combine genetic, quantitative genetics and also crop modeling. So crop model basically helps by estimating some yield based on information on the crop management, on the soil and the climate, and on phenotypic traits that describe the variety. And altogether they help with the yield production. So we did this using 3 control varieties that we know very well. And this model helps by calculating the average daily stress index for drought, nitrogen, cold, and heat. So we have a scenario of stress for these 4 abiotic stresses. And here, in this example, I'm going to keep the drought stress as the number of days of the fraction of transferable soil water below 60%. And this is a bit what we get. So this is basically the number of days of stress on the different environments, the bars, and these are the yields of the populations. The core collection, and in blue, and in green the 3 control varieties. And so you see there is a strong correlation between the drought stress and the loss in yield. And so when we look at one variety here, we have the stress index of the different environments, and we have the yield. And so we can actually have a slope, fit a linear model, and this slope is actually what we call the plasticity. So it's the way the genotype responds to drought. And this describes the sensitivity of the tolerance of the genotype for drought stress. And this is how we can calculate this impact of 1 day of stress on these commercial varieties as 0.4 quintal per hectare per day of stress. And so now if we do this fitting for all our population, we add 315 lines. we are able, thanks to genotyping, to identify one genomic region that controls this response to drought in this panel. So we sequence this, and we have the plot of the association between the markers here in the region and the p-value of the association to this plasticity to drought. And so we have this region that is the most associated, and different genes underneath, and especially an alternative oxidase that is very interesting. And this was already described to be involved in drought sensitivity in Arabidopsis, so this is very interesting. But what was very surprising to us is that when you compare this association between maintainer lines, you have this association, but when you just do it with restorer lines, you don't have a statistical association between the polymorphism in the region and the drought sensitivity. And here in our trial, we did everything in hybrid, so the maintainer lines, they were crossed to GMS or the CMS PEF1 sterility system, whereas the restorer lines, they were crossed to PET1, the classical PET1 sterility system. And so we think there is an interaction between this locus and the genotype of the cytoplasm or the mitochondria on the chloroplasts. So we wanted to find the gene, and so we used the Eliafen platform here. So it's a robot, outdoor robot that is automatically watering the plant and so managing drought stress scenarios. And so we did different experiments where we stop watering and we maintain it at 20% over a long time. And when we do this, we compare the expression of this alternative oxidase. We see that there are two copies. One only is expressed, so the other one is likely to be a pseudogene in seeds and in leaves. And when we check the impact in this condition on the yield in drought stress, we see that, of course, here it's stressed and here it's controlled. The drought stress impacts, of course, the 1,000 kernel weight, but also the genotype on the marker here. And what is interesting is that when here, it's when you have the allele of sensitivity, you have a strong impact of the seed on seed weight, but when you have the tolerance allele, you have no impact on the seed production in this condition. So this was very very interesting, and we wanted to find the way this genomic regions makes the sunflower tolerant to drought. And so we focused on this alternative oxidase. It's involved into reactive oxygen species production and then leaf senescence. And so we did a trial for on leaf senescence. So we looked at the leaf senescent leaf ratio in these conditions. So these are the conditions that we had before on the wild cytoplasm, the one for the seed production. So here we see again that the tolerance allele is having a less impact of drought stress, and here the sensitive allele, it has a higher impact on drought stress for leaf senescence. And when we do it on the PET1 cytoplasm, the same experiment, we actually observe that this allele has no more effect. So there is this confirms there is an interaction between the genotype of this genomic region on chromosome 3 and the genotype of the cytoplasm, likely the mitochondria, because the alternative oxidase is targeted to the mitochondria and is actually functioning in the mitochondria. So this is the most likely. And this also confirms that this is acting through the induction of leaf senescence by reactive oxygen species. So, so this is a synthesis on this approach. So basically, with the crop model, we are able to describe the different abiotic scenarios and describe stress indices. When we link it to yield in this multiple environmental trial, we can calculate a plasticity. So it's either tolerant or sensitive to the different abiotic stresses. And because we genotyped the population, we can do genome-wide association studies and identify this QTL, or the genomic regions controlling this tolerance. And now we published 9 regions like that control cold tolerance. Here I described one. In fact, we did many more that are still under validation procedure in the Sunrise project. And we are now looking for the genes that are controlling this, the phenotypic proxies, so to help breeding this trait, but by a much easier means than actually having this plasticity phenotype. So like leaf senescence at early stage, like I showed. And so this can help with marker-assisted selection, also looking for new alleles in the diversity panel that are presented, and of course help with the breeding and introducing these alleles into elite material. So I'm going to skip this perspective and challenges and really thank all the people. As you guess, there is a lot of people involved into this research. For the last part on the combination of genetics and crop modeling, I want to stress the participation of Pierre Casadebay on crop modeling that developed the crop model for sunflower, Brigitte Mangin, who's a statistician that just retired, and also Nicolas Blanchet on the Eliafen platform that ran all the experiments that we saw. And of course, everybody in the team at INRA, and our partners in Toulouse, in other teams that help with the genomic resources, that help with the field experimentation, etc. Thank you for your attention, and